to our next presenter, which is Shima. So Natasha's going to share your screen, and when you're ready for her to change slide, just say next and she'll change it for you.
Okay.
Can you guys see the slides?
Yes.
Great.
I think you can go ahead when you're ready.
I can't see it. Okay, I can't. Okay. Hello everyone, I'm Shima Hassan. My research is about in silico analysis of single nucleotide polymorphism, reporting the coding region of the RF6 that potentially causes autosomal dominant isolated non-syndromic cleft. Orofacial clefts are the most common congenital craniofacial abnormalities. Are you guys hearing me or?
It was a bit intermittent there, on and off. Go ahead, yeah.
Is my voice clear for all of you?
It is now. Go ahead.
Okay. Orofacial clefts are the most common congenital craniofacial abnormalities. Can happen as a part of the Mendelian syndrome or as isolated non-syndromic incident. Failure of union between any of the developmental facial processes leads to facial clefts. Can occur either unilaterally or bilaterally, and typically occur between the lateral incisor and the first premolar teeth. The depth of the cleft may vary from the lip to a complete cleft of the maxillary bone. And obviously, it negatively affects on the individual quality of life, hence need for better management. Next. Non-syndromic left lip is a common craniofacial malformation with a complex genetic component. Attempts at identifying susceptibility loci via family and case control studies have proved inconsistent. It's likely that initial prediction of the complex interaction involved in facial development were underestimated. The candidate gene list for cleft lip is getting longer and longer, and the need for impartial systemic screening technique to implicate or refute the inclusion of particular loci is apparent. Next.
Shima, we lost you there a bit.
I'm here, but I cannot see the screen for my presentation. The research aims to identify the variant in the interferon reverity factor 16 that may potentially cause isolated non-syndromic left lip. This is acquired through achieving the specific objective below: to identify the effects of variant on the structure and function of the RF6 protein, analysis of the 3D structure of mutated or mutated proteins, comparative analysis between the wild and mutated protein structure. Next. So, as I mentioned, my title of the research in silico analysis, that which means computerized analysis. So the method all was about using software and online tools. The first one is SIFT, and it's abbreviation for sorting intolerant from tolerant, and it's program that predict whether amino acid substitution affect protein function, whether it's tolerant or intolerant, so causing the disease. The polyphen 2 is basically looks like SIFT, but the difference between them that both of them have a scoring from 0 to 1. For the SIFT, the lesser the score, the more probability to cause a disease. And for polyphen 2, the greater the score, the more probability to causing a disease. Mutation tester predictor, it's slightly different from them. The result of it from it is the letter D, which abbreviation for disease. And the more it gives you a letter D, the more the probability of causing a disease. Next. Allele frequency is the relative frequency of an allele variant of gene at a particular locus in a population, which means we need a less or the most least frequent allele to say that it has a great probability to cause a disease. The I mutant is using a protein sequence or structure and something about thermodynamic change and decrease or increasing in the stability. It has score from 0 to 10. The greater the score, the more probability of causing a disease. FIRE2 was software for 3D model building. Use amino acid to build your 3D. Next, I took the model from the FIRE2 and visualized it in Chimera. It's 3D protein model reviewing, manipulating, analyzing, etc. The last one is HOPE, an abbreviation for have your or our protein explained, and it's just another software used in study for confirmatory purpose. It predicts the three-dimensional change that occur in protein. Next. So this is my result, and this is the positions with the highest scoring for probability causing, for disease causing. For SIFT, I get 0 for all of them. For polyphen, I get 1, which means probably damaging. And for SIFT, 0 means deleterious. For mutation tester prediction, I get double D for all of the position. And for mutant, I have a various score 6, 7, 8, 9, and which all of them indicate the stability decrease. The last column is something we called it clash point. That means in the three-dimensional form of the wild, I mean the normal and the mutated one. For the first position, for example, we have one clashing point, which means we have one interaction that we don't want it in the mutated. For the second, we have 1. For the third, for example, we have 11 clashing points that it doesn't occur in the normal or in the wild protein. Next. This is, for example, for the first position. In the left, we have the wild, and in the right, we have the mutated. And we can observe the difference between each of them. Next. So we can see a zooming picture of them. This is in zooming. We can see in the wide, it's a tyrosine in position 312. And the mutated, we have a histidine. And this gives us a slight difference. Next. This is the clash point I talked about before. We have one interaction, and in the right, we have a general matching of the wild and mutated RF6, and obviously we can see the difference between them. Next. So in conclusion, all of this mutation was found to be effectively altering the protein structure and function. Hence, it can be a major causative factor for autosomal dominant isolated non-syndromic left lip. Next. So I recommend it's necessary to conduct further genomic studies for orofacial clefts patients to discover the inheritance basis for the different pattern variants which affect the treatment option. Furthermore, screening for this specific variant is also initiated, not only to assess its prevalence among isolated non-syndromic left lip patients, but also to confirm its direct relation, hence application of different numerical measures for diagnostic and treatment purpose. Thank you for attention.
Thank you so much for your presentation. It was great. Do we have any questions from the audience? And while we wait for questions from the audience, I have a question. And now that you've done your in silico analysis and identified some variants, are you planning to do any functional lab work to confirm their effects?
Yes, sure. I'm planning for that. I'm just having some trouble, financial trouble. I'm finding the suitable lab for doing that, but it's my plan, my future plan for doing it.
Great, thank you. Do we have any more questions from the audience? Doesn't look like it, but we can give, oh, here we go. George says he likes a combination of tools. Not sure if I missed it, but have you used Dynamite? I think that's what that says.
I didn't use it. I just used the one which I mentioned. Not Dynamite.
Great, thank you. Do we have any other questions from the audience? If not, thank you so much for your presentation. No, wait, there's another comment. George says it's a protein-based variant interpretation server and he will send you the link.
Okay, thank you. Thanks in advance.
Thank you so much.